Good afternoon, everyone. My name is Marwan Abouzoud, Director of the Transplant Institute, and I want to welcome you to one of our Transplant Institute lecture series. We're very fortunate today to have with us Dr. Phil Halloran. For those of you in the transplant community know of Dr. Halloran, appreciate the level of leadership and knowledge and contributions he has made to the transplant community at large and to transplantation. Dr. Halloran started his education at the University of Toronto and subsequently went to the University of Alberta, where he directed the transplant unit and the nephrology division. He had spent some time doing research in immunology at the University of London as well, and spent some time there. And his expertise and interests were in immunology and antibody-mediated rejection. And this evolved over the years to trying to understand the mechanism of tissue injury, organ injury, and how they would translate into understanding of transplant rejection and graft injuries. And this basic knowledge evolved over time to the formation of a very large genomics and molecular research enterprise that culminated in him initiating and directing the Alberta Transplant Applied Genomics Center, which is world-renowned and quite innovative in trying to discover what are the molecular fingerprints of tissue injury, organ injury, and rejection. It is very difficult to narrow down in 1 or 2 minutes what Dr. Halloran stands for in transplantation. For us who read our literature, understand the value of the American Journal of Transplantation, and he was the founding editor of that journal, and now he is editor emeritus, which is a significant accomplishment. This is the highest rated index journal in our world of transplantation. He also has had several honors in his various roles, and one of the functions he occupied as a founding chairman of the Board of Trustees for the Rose Organ Transplant Research Foundation, which is a Swiss-based charity, and he's also an officer of the Order of Canada and a fellow of the Royal Society of Canada. And it is difficult to humanize somebody with superior achievements of that scale. When I tried to narrow down some of the items on his CV, I came to appreciate that he was invited to over 580 invited lectures, over 330 peer-reviewed journals, and with a CV that weighs about 10 times my CV, even if I use heavy paper. So with that, it's my pleasure to introduce you Dr. Halloran, who's going to be talking to us about the molecular microscope and how that pertains to transplant and tissue injury.
Thank you very much. Thank you for that generous introduction. I haven't been here for about 70 years. When I was a very small boy, well, it was actually a little less than that, but my sister and I had eczema, and we came here from Hamilton in Ontario because the Henry Ford Hospital was that brilliant place down in Detroit, which was the center of the universe at that time. So it's been a long time since I've been here, and it's a real pleasure to be back. I'm very pleased to see what you've done with the Transplant Institute, and I think that this is a credit to the way people are working here together, which is a credit to you and to all of your colleagues in leadership here. So well done. So what I'll be talking about is how to bring molecules into the clinic. And my disclosure is that our center has created a system which brings molecules into a practical way that can be read in biopsies. And we've licensed this to Thermo Fisher. And we're setting up centers that can do this. It can't be done as a kit. It has to be done as a center. So we set up the first center in Portland, Oregon to provide this as a service, and that's in Kashi Laboratories. And if you're interested in finding more about these services, you could contact Hal Gibson, and I could give you his contacts. At any rate, the molecular microscope project brings all of transplantation together because we've got one platform that we're going to be able to read livers, kidneys, hearts, lungs, and not just read them, but actually tell you about the science of the injury and rejection phenotypes that are going on in the tissues, which is really where I come into the picture. I want to understand the disease states. So in many ways, once we get rejection under control, then what is emerging as the important signal of tissue injury, which can only be read in the molecules. You cannot see this in histology, the way tissues respond to wounding. And the interesting thing is that tissues have liver, kidney, heart, lung. They have certain principles, and they actually have rather simple ways of responding, and they all seem to respond in similar ways. You can actually see this injury signal in cancer as well. So we're starting to learn some things about the biology underlying injury. Remember, the patient came to us for the parenchyma. The adaptive immune response is of incidental interest, but it's parenchymal function which we're trying to understand, and how the tissue responds to wounding. So that's actually going to be the next big theme we develop. So we'll talk about why we're doing this, and then we'll talk about the system and show you what we're learning about the molecules. I know you're not molecule people, so I'm showing you the molecules only to say this is the herd of little creatures that we're working with and how we're turning it into a diagnostic system, but I hope not to overwhelm you with molecules. I'll show you how this relates to the conventional system of histology. I worked my whole life with histology, and now I'm working with molecules and trying to see how these work together, and not competitively. And then we'll show you a little bit about what we're doing in heart and lung, and we're getting started in liver. So the unmet need is to get precision and accuracy in clinical assessments. Now, when you look at, this is accurate and precise. This is accurate, but it's not precise. This is precise, but it's not accurate. It's called biased. It's like saying Mexicans are rapists. You've got a bias, and you can be as accurate as you want about determining who's a Mexican, but it's not going to help you how to find a rapist if you're dealing with a bias. And that's what a bias looks like. So precision can't stand bias. So what we want is actually to go back here. This is the worst case where you're both imprecise and biased, and a lot of our clinical things are acting like that. So it isn't just precision medicine. It's precision, accurate medicine that we're headed toward. Now, precision should involve statistical assessments which use our quantitative numbers. Don't dichotomize things which are discontinuous. If the data suggests there's an 80% probability, it doesn't become dichotomous like 0 and 1. It's 80%, and retain the numbers. So predictions, and I'm a big fan of Nate Silver, regardless of his performance at predicting elections, I'm still a fan of the fact that predictions, like clinical assessments, should be probabilistic, consensus-seeking, updatable, and consider the prior probabilities. And that's, if you know about Reverend Bayes, that's Bayesian. So that's the way we should be thinking clinically. And your brain actually works that way, but diagnostic systems often try to simplify this into 0 and 1, and it's not 0 and 1. So the clinician needs to know what the rejection status is. The clinician needs also to know what the injury status is. Is there recent parenchymal injury? Is there irreversible injury? And is it going to progress? Those are the elements that ultimately we're going to work on after we've solved the problems of rejection. Now, the problems with empirical approaches, and we'll look at histology as one of these approaches, that they're currently, the conventional approaches are empirical. They were derived from the ground up by clinician opinion. They're based on non-specific features in particular circumstances, and then you apply rules. We call them guidelines. And experts interpret the guidelines, and they don't always. They say, it depends. And that's not the way computers work. Computers don't come back at you and say, well, it depends. No, there's an algorithm, and you run the algorithm. So if it depends on something, put it in the algorithm. It shouldn't depend. At any rate, very often conventional systems go to 0 and 1. It's cancer. It's not. There's poor assessment of recent injury. So recent injury is often a player in phenotyping, and we can't assess that. And the intra-observer variability is huge. For example, observers tend to be good at normal versus abnormal, but they're not very good once you get into what kinds of abnormal. And actually, your grandmother's probably pretty good at normal versus abnormal. Rejection versus no rejection, you often don't need a microscope. This one's blue and this one's pink. But that's not what we're looking at. We're looking at the whole spectrum in between and how to assess that. So once you're in the abnormal, pathologists in kidney transplant looking at T-cell mediated rejection, if one diagnoses T-cell mediated rejection in kidney transplantation, a second will agree 50% of the time. In heart transplantation, that's 28% of the time. In lung transplantation, it's a coin toss. Lung transplant histopathology is probably essentially worthless. And that you can see that in the inter-observer agreement. These are kappa values. And this is actually published. And then in liver transplant, we just don't know. So that's actually what we're trying to fix. You can do better than this. Our expectation is that when we look at molecular readings, we will agree with the conventional phenotype to the limits of the noise in the conventional phenotype. So if the inter-observer agreement is 50%, we'll agree with the conventional phenotype 50% of the time. Not 100%. You can't agree 100% if the noise in the system is 50%. Or you'll be guilty of the sin of overfitting. If you see that, if you're better than the noise in the system, you have overfit the data and it's not going to work. So now we have an expectation and we see the unmet need, and so our system goes like this. We are currently doing research protocols, clinicaltrials.gov, so we get a piece of tissue in RNA later from a consented human from Vienna or Melbourne, Australia, or Richmond, Virginia. It goes into, so the biopsy goes into RNA later immediately. In the kidneys, we get 3 millimeters of a core. It comes to us by FedEx. Overnight, then we go into the RNA extraction and cleanup, quality control, labeling, hybridization to a chip, and then a wash and stain and read the chip and generate an automated report, and that takes about 24 hours. So overnight to us, 24 hours, and that's an automated report, and then we send that back to our clinicians. So the molecular microscope system is a central diagnostic system it could be centralized in one center, like your center, but it has to be set up and calibrated in the center that is run. So it's not a kit. So it's a centralized diagnostic system that uses microarrays to measure transcript changes in biopsies that have been stabilized in RNA later and shipped at room temperature by FedEx, just in basically mail. And so what we do is we get the readout from the chip, we apply equations which we've worked out over a large number of years, which turn that chip into a diagnostic system which says what the disease and injury states are in that biopsy. And our system decides whether you have any rejection, whether it's T-cell mediated or antibody mediated, and we get an estimate of whether that's due to non-adherence. And then in addition, we look at the parenchymal wounding features, and so that's taken off the chip reading. And these are the centers that are working with us, and these are the clinicaltrials.gov numbers of the studies that I'm talking about for kidney, heart, and lung, and interliver protocol is under development, and we'd love to have you involved in interliver. Currently what we've done is establish this service laboratory. We, being Thermo Fisher, has running its license in this service laboratory at Cashy Labs in Portland, Oregon, and they will be doing this system under our supervision, and there'll be other centers doing this, and we're now talking to 2 European centers about setting the system up for European customers. And this is the team that's worked on it in Edmonton, and that's Zara Cashy in Portland. These are my sons who work on the project. Brendan's a gastroenterologist, and he works on ulcerative colitis with us, and Kieran is a lung transplant doc, and he has done many of the lung biopsies that we're incorporating into the system. And these are some of the, I just like to always pay tribute to the patients who, all of this is based on consented patients. So we've got several thousand pieces of human tissue, but it's always from a person who has committed themselves to the study. Now, what's the sample size? So the sample size is considerably less than is required for histology. So in kidney, we get about 3 millimeters of a core on average. We can make an adjustment for medulla, so we can read medulla. Histology can't, but we can. And then in heart, we're getting one endomyocardial biopsy bite. They usually take 5, but we would like probably 2 bites, but probably will eliminate histology. And lung will almost certainly eliminate histology, and we'll probably get 2 bites. Currently, the IRB only permits 1 transbronchial biopsy. We're actually moving to try to read mucosal biopsies in lungs so we can replace the transbronchial biopsy ultimately, and I'll tell you about that. So in liver, the amount of tissue remains to be determined, but I suspect we can read about the same amount of tissue as we get from kidney, about a 3 millimeter bite from a core. And the big advantage is that we get a beautiful profile of injury. We've read the injury profile because in kidney we get GFR in recently transplanted kidney is histologically pristine and gets a massive injury response, even live donor kidneys. And so that's where we've learned to read the injury response in tissue which has no histologic signal. So we've taken that into the other organs. It's interesting that the injury response basically crosses organ barriers very well. Not surprisingly, because it's often, the pathways here are the pathways of cancer. Now, developing this in kidney, we started off, basically this is all reviewed in Nature Reviews Nephrology that came out last year. So I'll take you through the system. I'll show you some molecules. Don't worry about it. You can sleep for about 2 minutes. I'll just show you the molecular phenotype. And this is very robust. We do it in one population. We take the same molecule. We do it in another. So this is consistent now with probably about 2,000 kidney biopsies. And so what we do is we do algorithms like this. This is an algorithm which we say T-cell mediated rejection versus everything else in the biopsy population. And that algorithm generates molecules which are basically related to T-cell recognition and activation and antigen presentation cell activation. So this is reading a synapse in the tissue. So you're getting molecules from the antigen presenting cell, ADAMDEC1, SLAM-F8, and molecules from the activated T cell, interferon gamma and CTLA4. And those are the transcripts at the very top of this. This is the p-value for their association with T-cell mediated rejection. And this is 10 to the minus 45. And this is the fold change. This is called a volcano plot. So really we're looking at the p-value of the association. So that's the molecular phenotype most associated with T-cell mediated rejection. And that is the same phenotype we see in heart transplants and kidney transplants and now lung transplants. So what we're actually seeing, we think, is the effector T cell has crossed the endothelium, is engaging an antigen-presenting cell in the parenchyma of the tissue, in the interstitium, and that's creating an injury response in the local tissue, and that's what T-cell mediated rejection looks like. I won't go through all the molecular biology of this, but it looks to us from experimental models like the rejection is actually not a cytotoxic event, even though it's mediated by a T cell that has cytotoxic molecules. Now, the one thing about T-cell mediated rejection is that after 5 years, this is the linear scale, after 5 years, we very seldom see T-cell mediated rejection. This axis is molecular T-cell mediated rejection in the biopsy. The red color is histologic T-cell mediated rejection in the biopsy. And after 5 years, we just stop seeing it. And basically, after 10 years, it's very seldom seen in kidney transplants, even in non-adherent patients. Now, antibody-mediated rejection continues indefinitely, even after 30 years. Why is that? So why does the T-cell mediated rejection cease to occur after a certain period of time? So we've been asking ourselves this, but also watching the cancer literature. It turns out that the T-cell is wired to engage to go to certain phenotypes. If the antigen disappears, it goes to memory. If the antigen persists, it goes to exhaustion, and that exhaustion phenotype is the story that's emerged as the hottest story in all of cancer. Is that exhausted T cells that's mediated by 3 mechanisms. There's the CTLA4 mechanism, the PD-1 mechanism, and the ICOS mechanism. And interestingly, in the T cell mediated rejection phenotype, we see that the rejection is operating with the brakes engaged. It's working against the brakes because these inhibitors, these checkpoints, are actually expressed in the T-cell mediated rejection phenotype. Now that raised the question, does some T-cell mediated rejection therefore not need to be treated because it's exhausted? And so we see in protocol biopsies T-cell mediated rejection histologically and molecularly, but it's probably a state of exhausted T cells. It's created a phenotype, it's a stable state, it may not need to be treated. In fact, it may be counterproductive to treat it. But that's a question we can now answer. And then what would happen if we treated someone who's used the T-cell exhaustion mechanism to achieve stability, and if they were treated with a checkpoint inhibitor, what would happen? And you've probably seen some of this literature. I've been quite excited about this. We're actually working in melanoma as well, so I've been quite excited about relating human T-cell mediated rejection in the kidney to the phenotype of the people getting checkpoint inhibitors in their melanoma. And so the first really good case of this was published last year in the New England Journal. I think you may have seen this. And basically, this is a lady 28 years post-kidney transplantation with a stable kidney transplant. Basically, T-cell mediated rejection just doesn't occur after 10 years. She got treated with a checkpoint inhibitor, and she blew the kidney away. Her squamous cell carcinoma regressed, but it showed that she was probably using her checkpoints to stabilize her T-cell response. And when she received the checkpoint inhibitor, she blew the kidney away with uncontrollable T-cell mediated rejection. No antibody. So that was a case that established the principle, in my mind anyway, that the patients in the clinic are probably using the checkpoints to stabilize themselves for T-cell mediated rejection. So it raises the question also, you're seeing patients out there who are getting squamous cell carcinomas the way I'm seeing them, and we've given them the squamous cell carcinoma, so should they be getting checkpoint inhibitors? And we're having to face this question with patients, and you'll be facing it more and more as the checkpoint inhibitors spread into other kinds of cancer. Should we be treating? And I've actually had to talk to patients about this, and it's a really difficult choice for them and for us. Because we gave them the squamous cell carcinoma, we have a special interest in this. So antibody-mediated rejection, just to show you the landscape of antibody-mediated rejection. When we do antibody-mediated rejection versus everything else, we say, what are the molecules associated with this phenotype? What we always get are the molecules associated with endothelium, with interferon gamma-inducible genes, and with NK cells. So it looks like the population, the antibody-mediated rejection does look like it's an antibody-dependent cell-mediated cytotoxicity problem, at least to some extent, not a complement problem. It looks like NK cells are killing the endothelium by recognizing it through their FC receptors, engaged to the antibody bound to the endothelium. So that's our model for how antibody-mediated rejection works. So that suggests that there's a similar synapse, only this time the synapse is between the NK cell, the FC receptor, the FC portion of the antibody, and the endothelium. So that synapse is probably activating both of those cells. Whether that's cytotoxic or cytokine driven, we're not sure. And you can't model that in animals because in animals, experimental animals don't have the NK system that humans do. They don't have NK receptors which are activated with a total activation machinery, and mice don't have that. So that you can only do this in humans and in higher primates, but you can't. Baboons are a very difficult place to do experimental research, but they're using the same mechanism, it seems. When we look at antibody-mediated rejection, we actually get two types. There's type 1, which is you transplanted the patient against a DSA and they get early antibody-mediated rejection. And there's type 2, which is the much more common, which is the naturally occurring de novo DSA. And we just showed in JSON that type 1 has a much better survival than type 2. So the average case is much more refractory to drugs than the ones that you transplant and you get an early DSA. So this is basically one of the fundamental rules. And so if you're doing clinical trials of type 1, don't expect the results to extrapolate into type 2 because it's a different situation. So now we have these three phenotypes, and we can pick the top genes in each of these phenotypes and call them rejection-associated transcripts, or RATs, and we'll show you how we use that to create tests for the other organs. And we take these molecules and we put them into mathematical classifiers by machine learning, and we translate this into a report. And this is what our clinicians are getting when they send us bits of human. So this is the report that we'll send out. We'll say how much inflammation there is. That's the disturbance. the disturbance in the force, how much acute kidney injury there is. In this case, there's a lot of acute kidney injury, even though this is 8 years post-transplant. And this case, a lot of atrophy fibrosis, well, a moderate amount. And then the rejection molecules are up, the antibody-mediated rejection molecules are up, but the T-cell mediated rejection score is basically 0. So this is what antibody-mediated rejection looks like. Now here's what we do to make this precise. We take that patient and we show you that patient in nearest neighbors, so that we're looking at that patient against the memory that we have of all the patients that we've done, and we're able to show you what other patients have done that are like this patient. So that's nearest neighbors technology. It's done by distance between the measurements in these biopsies. That's your patient, and these are the other patients in the data. The color is the diagnosis histologically. The position is the diagnosis molecularly. You can see many of these have been called antibody-mediated rejection, but some of them haven't. And this is the noise in the histology classification of antibody-mediated rejection, which I'll show you. And what's happened to these patients? So these are the nearest neighbors. The nearest neighbors of this patient had a 3-year graft survival of 43%. That's the way you can use the memory of the system to highlight what's happened to patients like this, the nearest neighbors technology. So we've used this in a prospective study to ask our clinicians to say how do they like getting these measurements. And this paper is in AJT online, and we found that this is a prospective study to look at 500 consecutive biopsies sent to us from consented patients in North America and Europe. We're able to process 96% of these biopsies, and the 4% we couldn't process basically because this is the first time they'd ever done it, and some of the centers made mistakes with processing the specimen. They sent us too little tissue, or they sent us only blood, or they accidentally mishandled the tissue and didn't get it into RNA later. Once it's an RNA later, it's very stable, but it has to go into RNA later like that, and then you're done. But you can't go and have coffee and leave it on the bench. So we processed 96%. When the agreement with histology showed us 77% roughly agreement. That's called balanced accuracy. It's the average of the specificity plus the sensitivity divided by 2. And the balanced accuracy reflects basically the noise in histology. We can't agree with histology better than histology agrees with itself. And when the clinicians were asked about our readings versus histology, they said that our readings were a better reflection of what they felt was going on in the patients than histology was. So in 81% of the feedback forms, the clinicians reported that the MMDX readings would increase their confidence in management compared to the absence of those readings. So this is a research study, but it was very encouraging to us. When the two readings disagreed, histology and molecular, the molecular reading reports were said by the clinicians to potentially be capable of triggering a change in anti-rejection therapy and maintenance immunosuppression, and triggering other investigations, so that they felt that this would be useful in a lot of biopsies. And the interesting thing is clinicians frequently disagree with their own histology assessment. And that perhaps wasn't a surprise to me if I'd thought about it, but it was a surprise to find out how widespread that is. Now, so now we can do many, many measurements. We can run many equations on the biopsy. And it's like getting, each equation is a different opinion. It's like having a different pathologist read the slides. So now we actually do 84 different mathematical algorithms to look at rejection alone in the biopsy. And now how do you put 84 algorithms together? So now you've got 84 different assessments of the rejection phenotype. And so we have a method called archetypal analysis, which has been derived from the marketing industry, but what it does is it puts together multiple estimates and says, how many entities do you have in this data? And it said, we have 6 entities. We have, and this is basically called an unsupervised analysis, Among the biopsies, there's no rejection, T-cell mediated rejection, mixed, and 3 stages of antibody-mediated rejection. And then we summate those to get a total antibody-mediated rejection score. And then we are starting the analysis of injury in the same way and finding out how many different injury phenotypes are there in a tissue. And we're not there yet, but I'll just show you what this analysis looks like for kidneys. And so now we're actually trying to get to the ultimate molecular readout of that piece of tissue. So what this is, we do a method for any of you with a little math background. This is tenfold cross-validation, and so the machine learning goes through multiple tenfold cross-validation loops to take an algorithm and create an estimate, which is the estimate for one of these different equations. And so this is how the 84 equations are generated. And when we do that, we find the biopsies distribute in principal component analysis like this. So this is a way of getting the distribution of the rejection phenotypes in a human transplant population. So by principal component analysis, this means the main deviation is from no rejection toward rejection, and then it separates in principal component 2 into antibody-mediated rejection and T-cell mediated rejection. And then it separates a little bit out this way toward mixed rejection. Now if we take that diagram and we turn it toward you, so now you're looking at the axes are still principal component 2, but now you're looking down the barrel, we see that there's actually a lot of divergence in a third dimension, and that divergence in the third dimension we were very interested in. So this is antibody-mediated rejection is diverse, T-cell mediated rejection is homogeneous. So now we look in the third dimension, and the way we do this is with the archetype analysis, and the archetype analysis actually looks at this and creates these archetypes for us. I don't need to take you through that except to show you the output. It says this is the archetypes. There are the 6 archetypes. There's no rejection, T-cell mediated rejection, mixed, then early stage, fully developed, and late stage antibody-mediated rejection. And that's principal component 2 versus 1. If you take it and make it 2 versus 3, you see that the divergence across antibody-mediated rejection is that there's an early stage, a fully developed stage, and a late stage, and they're different. They're molecularly different. And so now we can actually assess antibody-mediated rejection stage, and it's actually reassuring that T-cell mediated rejection only has one phenotype. It's either there or it's not. It's quantitatively different, but it doesn't have a divergence of phenotype. There's no such thing as chronic T-cell mediated rejection. It's just T-cell mediated rejection or not. So these differ over time, and time up of the biopsy after transplant is a huge piece of information. And we don't put it into the computer, but the computer extracts it because the phenotypes differ over time, the molecules differ over time. So non-rejection is common in the very earliest biopsies. You think there might be rejection, but mostly it's injury. Interestingly, some of these biopsies are read histologically as rejection because histology inflammation caused by injury looks very much like T-cell mediated rejection. But basically, non-rejection becomes less frequent over time. The first rejection phenotype is early antibody-mediated rejection in those DSA-positive patients that we transplanted and we didn't get right, and they got an early episode of antibody-mediated rejection. So that's early in time, and it's also early stage. Then there's a second peak of early stage antibody-mediated rejection caused by de novo antibody responses. Then there's fully developed antibody-mediated rejection. This occurs at about 200 days. This is about 1,800 days or 5 years at that peak. Then what happens in the late stage is the emergence after 10 years of late stage antibody-mediated rejection dominated by the basement membrane changes, double contours. So that's the natural history of the antibody-mediated rejection problem in a wild-type human transplant population, unselected. T-cell mediated rejection peaks in the second half of the first year and then has another hump in the non-adherence period. So when we look at this, that is the non-adherence problem. And the 3 phenotypes of non-adherence are mixed rejection, early stage antibody-mediated rejection occurring in the second year, third year, and T-cell-mediated rejection occurring in the second and third year. So that is the non-adherence phenotypes. And when we look at survival, that's just some raw data from those scores distributed over time. This is the actuarial survival curves of the patients from the time of biopsy. So the best thing is to not have rejection. The second best thing is to have T-cell-mediated rejection, but having an antibody-mediated rejection phenotype of fully developed or late is very bad for you. If you have early stage, it falls on a curve which is delayed by about 2 years from the fully developed and late stage curves. So that, again, is helping us get a feel for the natural history of the disease state. Very important as we now come to support clinical trials in antibody-mediated rejection. So now this is the report we'll be sending you from the service center when you send in biopsies. You'll be getting a report that looks like this, and I'll just walk you through this. These lines show you the results of injury classifiers. These are some rejection classifiers, but this is the analysis that I just showed you. And this patient has fully developed or intermediately fully developed antibody-mediated rejection, so that they've got pretty well the full house. They're still on the early side of full house. They don't have any T-cell-mediated rejection. So this is principal component 1 versus 2. This is principal component 2 versus 3. So that's where they are. This patient has mixed T-cell-mediated rejection. And this phenotype is often associated with non-adherence, so that's where they are. And that's where they are in space. And if we look at page 2 of the report, we have a classifier we're working on, and this is under review at AJT. And that is a classifier based on underhyalinosis. And that is our key to telling which biopsies have non-adherence. If they don't have enough hyalinosis, then in certain time periods, that indicates that they're non-adherent. And we're using that as a way of developing a classifier, and this patient is very non-adherent. He's very much less molecular changes related to hyalinosis than were expected for its time post-biopsy. So that's our key to giving you a non-adherence phenotype. Now, we were very interested, spent most of my career working with histology phenotypes. Now, histology and molecules are actually looking at an unknown phenotype. The truth is unknown. We're actually interrogating the truth from different platforms. So we make the assumption that molecules are right, because they don't have inter-observer variation, etc. So if we make that assumption, we can then look at histology phenotypes and see how right are histology phenotypes. So this is instructive. We can now look, and I won't take you through this table, basically molecular histology discrepancies. Overall, the discrepancy rate is 39%. If we look at all the things we're measuring and all the things that histology are measuring, which nominally is the same thing, we get a 39% discrepancy rate. In biopsies where histology says there's no rejection, we have 16% discrepancy rate. In biopsies where histology says there's rejection, we have a 72% discrepancy rate. Now, we've seen the discrepancies are of 3 types. Now, remember, we're looking at with the world's leading transplant centers, and we're just asking, what does your center call this biopsy? We're not recording a specific pathologist read. It's not a research read. It's standard of care. And pathologists don't like this because they say you should be sending them to Bob Coleman at Mass General. No, we're actually interested in the standard of care. So the standard of care, basically we find there are 3 types of errors, and they're all very common. There's pathologist error, who's actually recording the lesions, but he's actually making a diagnosis that isn't supported by the lesions. And there's BAMF error, which is, BAMF has actually got rules in it which we've said are wrong, but they persist in BAMF. And then there is BAMF correct, and this is the histology system basically is limited by using non-specific lesions, so there's an inbuilt error in the BAMF correct. So when we look at, for example, the nearest neighbors, we've turned this upside down. This is the T-cell mediated rejection area, the mixed area, the antibody mediated rejection area, and the no rejection area. So now we've taken cases that look like discrepancies. When we look at discrepancies here in the molecular T-cell mediated rejection, it's been called non-rejection. And we found that this type of case is 7 of these 43 cases were pathologist errors. They recorded lesions, they made the wrong diagnosis for the lesions. 24 were Banff errors. It was because of erroneous rules that Banff continues to have in it, which we've shown them but haven't been changed. And 12 were actually Banff correct, but Banff says this is not rejection, and it is molecularly. And then as we go through the other areas, this is the antibody-mediated rejection area. These are called mixed, and that's because the T-cell mediated rejection features are actually tubulitis and interstitial infiltrate, and they occur in any form of inflammatory nephron injury. They occur in glomerulonephritis, so that's built in that some cases of antibody-mediated rejection will have the tubulitis and interstitial infiltrate. So does glomerulonephritis, but Banff doesn't take that into account, and it calls these mixed, and these patients will be getting treated for T-cell mediated rejection they don't have. These are cases called borderline. Why? Because Banff will not allow you to diagnose antibody-mediated rejection if you don't have DSA, but the standard of care DSA often misses a DSA. So they can't be called antibody-mediated rejection. These are cases called T-cell mediated rejection in antibody-mediated rejection. Again, because they have T-cell mediated rejection tubulitis lesions, just because that's part of nephron injury. These are cases would have no rejection, but molecularly they have no rejection, but histologically they sometimes get called rejection. Again, there are the 3 types of error in that. There's the error caused by misreading Banff lesions, by erroneous Banff rules, and there's an inherent error in using nonspecific lesions histologically. And so we think that that's basically the reason for a lot of these discrepancies. So we have overall, in 161 discrepancies we've analyzed, we have 64 pathologist errors, 38 erroneous Banff rules being used, and 59 which are inherent in the Banff system. Basically, it's the non-specificity of Banff, the inability to resolve the disease states. And why do we think that molecules are better? It's because when we have something external, like survival after the biopsy, the molecules always beat histology. So this is just the statistics that we've used in this paper. This paper is in JCI Insight. It isn't yet in Online Early, but it will be in JCI Insight. And when we look at those cases where the Banff rule says that if you don't have DSA, you don't have antibody-mediated rejection, no matter how typical your lesions are. And so we've looked at the cases that have molecular antibody-mediated rejection, whether they're DSA positive or not, by local standard of care, and they're both at high risk. And that tells us that basically the rule that you cannot diagnose typical antibody-mediated rejection if you don't have DSA is wrong, because sometimes DSA can't be detected for a number of reasons by the local standard of care. Now we've taken this into hearts, and we're having a lot of fun in hearts, and we had the circulation paper just came out, and there's a paper in Journal of Heart and Lung Transplantation which is in press. I don't think the PDF is out yet, but we can send it to you if you would send me a request. So basically we're talking about the endomyocardial biopsy, which is the standard of care for diagnosing heart rejection over the last 50 years or so. And the circulation paper just said, if we look at hearts and look at the molecular landscape of heart antibody-mediated rejection, we get a landscape which is just about the same as kidney antibody-mediated rejection. So the same molecules are used. So in this paper, we showed that there's CXCL11, CCL4, there's the CD16 Fc receptor, there's CCL4 again, PLA1A, which is the top molecule in kidney. This is basically the same phenotype as kidneys with antibody-mediated rejection. So that was very encouraging. It encouraged us that the principle of moving molecules between the organs for rejection purposes was suitable, that the molecules are basically agnostic to the organ that they're in. Even the endothelial molecules of the heart microcirculation have reacted similarly in many cases to the molecules of, like ROBO4 is up in heart antibody-mediated rejection and in kidney, and it's very much a microcirculation molecule. So this allowed us to create a diagnostic system in an unsupervised way by asking the computer, say, these are the molecules of rejection, the rejection-associated transcripts. Now look at the heart microarray results and show us which of the hearts have rejection. So it's basically using what's called an unsupervised analysis. And when we did that, we were able to get three archetypal classes: no rejection, T-cell mediated rejection, antibody mediated rejection. And if we do that in kidney, we get antibody-mediated rejection, T-cell-mediated rejection, and no rejection with the rejection-associated molecules. If we do it in heart, we get the same three classes. And these correlate with the histologic classes, but show there's enormous error in the histologic classes. And if we do it in lungs, we're getting the same three classes. Now, we're at early stages in the lung, but we are able, I'll show you the lung results. So this is an unsupervised analysis of all the heart transplants using the kidney rejection-associated molecules. And this case, this is from, I can't remember, this may be from Cedars-Sinai, John Kabashigawa's cases, and basically this is where you'd want your heart transplant to be. It basically, that's normal heart tissue. This person is in trouble. This person has relatively severe antibody-mediated rejection. And we've got a number of other measurements we can provide. For example, the heart transcripts have been depressed as a response to injury. The heart injury transplants have been increased. So there's a lot of parenchymal injury in this as well as antibody-mediated rejection. Now we've taken this into lungs, and the lung is very difficult because the phenotypes, for those of you who are clinicians in lungs, my son, who's the lung transplant physician, has told me how difficult it is. And the histology provides almost no useful information, and we can now see why. And they've got pneumonia, they've got reflux, they've got so many phenotypes operating. But it's the biggest unmet need because the biopsy is incredibly invasive, can't be used in a lot of the sickest patients, and gives them terrible information, so that they need diagnostic help. So what we've done here is basically we've been reading lung biopsies, importing the kidney transcripts. So this is the transbronchial biopsy. We've started a project where we're looking at the mucosal biopsies from the third bronchial bifurcation. And we're going ultimately to compare these two platforms. This is what we're going after first. We're using 2 bites, and many centers are using 10 for histology, and every bite is a risk of pneumothorax and bleeding. So we think we can read lungs with 2 bites at the max. We're actually only getting 1 bite right now. But ultimately, we'd like to move it to the third bifurcation mucosal biopsy because it's donor tissue which should be rejecting. And this is one of our first reports. This is one of Kieran's patients, and this patient seems to have T-cell mediated rejection. All the biopsies in this region have high expression of T-cell molecules. The interesting thing is when we look at the distribution of the histologic diagnoses of T-cell mediated rejection in lungs, they're up in this region. And this isn't where the T-cell molecules are. So we think that most diagnoses of T-cell mediated rejection in lung are actually wrong. So I think I'll knock off at this point. There's a lot of new points I could go through with you. One is, so we're doing real-time assessments, and this ordinarily will take from your biopsy needle to the report 48 hours. We're looking at technical and biological replication, which is very high. We're looking at tissue heterogeneity and how to build in measures of the heterogeneity of the tissue. So for lungs, we're building in a measure of how much alveolus is there. And that's done by surfactant expression in the biopsy. So we can get measures of tissue heterogeneity. We have to understand that molecules are non-specific. Nature uses molecules in different ways. Even if they're highly associated with T-cell mediated rejection, they're sometimes used in injury. So that's why you need machine learning. You can't just measure molecules. You need machine learning classifiers. And that's why we're doing archetypal analysis. we're also looking at FFPE tissue. And FFPE tissue can be read. You can generate a system for doing it in paraffin-embedded tissue, but the paraffin, the formalin fixation profoundly damages RNA and creates false positives and false negatives. So you're going to be left with some noise. So we can set up a system if you want it, but we don't think that's the way to go. We think that snap frozen or RNA later RNA is the best way, and FFPE is very much a secondary way if you can't get fresh frozen tissue or RNA later tissue. And that's where we are. So thanks very much for your attention. I've really enjoyed my visit here. I'd like to congratulate you and all the members of the Henry Ford Transplant Institute. I think it's exemplary in the way it's functioning, and you're functioning much better and you're much more successful than other attempts to form transplant institutes. So keep up the good work.
Thank you. Thank you, Dr. Halloran. The floor is open for questions. Anita?
Two questions actually. One is for the Banff borderline rejection that is read by the pathologist. The physician really does not know what to do. Are these exhausted T cells, or are these truly either Tregs or they are the T cells with injury?
So we looked at FOX-positive T cells. FOX3-positive T cells are present in damaged tissue. They go into damaged tissue. They don't tell you anything else other than the tissue's damaged. So it's a strategy the immune system has for stabilizing damaged tissue, but it's not interesting. You already know the tissue's damaged. So borderline turns out to be about 10% T-cell mediated rejection, 20% antibody mediated rejection, and about 70% tissue injury. So borderline ceases to exist because it then turns into probabilities. And the probabilities for most cases will be low, but some cases are I2, T1, and they haven't met the criteria. And they have it, except T1 has a kappa value. The tubulitis reading has a kappa value of 0.3. So basically, another pathologist would look at it and say it's T-cell mediated rejection. So it's basically the problems of using a dichotomous or categorical system is losing data, and it's not reproducible. So that borderline is sometimes typical T-cell mediated rejection, which didn't make the criteria by tubulitis score, but most of the time, and often it's antibody mediated rejection for some of those reasons I showed you. There isn't DSA, so the pathologist can't call it, because the rule is if it's not DSA, you can't call it. And I know transplant nephrologists have said, well, we've got to fix this because my insurance company won't pay me to treat these people because it doesn't, et cetera. So this is a case where you've got to get into the data and fix some of these problems which are constitutive. So that's what borderline is. There's a category called suspicious for antibody-mediated rejection, which is the same thing. These disappear when you do it by mathematics rather than opinion and categories. My sister had a breast lump. It was very small, and it was looked at by 3 pathologists. 2 said it's cancer, and 1 said it wasn't. Did my sister have cancer? Well, who knows? Because you have to start thinking of these as probabilistic. So if on a probabilistic scale, you'd then make a decision based on the probabilities, the cases of making a negative error, the damage of doing a positive error. But you should know that it's not cancer versus no cancer. It's a probabilistic estimate based on experts. And actually, we should be doing better than this. This is where precision medicine, precision accurate medicine should be going.
So the second question, what about exosomes? Exosomes can carry genetic material and carry just protein material. How would you reconcile the role of exosomes versus the synapse that you showed?
Yeah, so this information may or may not be represented in exosomes. That's a question to be asked. It potentially could be represented in exosomes. I wouldn't be surprised if the technology can be developed to actually extract this information from the exosomes. So far, it hasn't been. Body fluids generally cannot have this information. it won't be represented. So the body fluid information like cell-free DNA has to be non-specific. So that to get specific information right now, you have to go to the biopsy. But believe me, all of us are looking for opportunities to get specific information out of body fluids to not have to do invasive things. But right now, the stage is set to be less invasive by taking a kidney biopsy, which is only 1 core or half of core, or taking a smaller liver biopsy, taking only 2 transbronchial biopsies instead of 10. The stage is set to make this safer and more effective now by using these technologies and reducing the tissue requirements, but ultimately going to something which is not requiring a needle sticking into a human.
What are you recommending for your patients with squamous cell carcinoma?
So, I had this conversation with a patient I've looked after for 30 years. Let's call him James, because that's his name. So James and I gamed this. And so he's not gone invasive yet, but he's had multiple squamous cell carcinomas, and he's a time bomb. And I'm saying to James, there are some things we could do, and he says, I'm not putting my kidney at risk. So this is going to be an existential choice that the patients make with you, but there's also the cost. It's $200,000 for some of these treatments. So if it goes invasive and their lives are at risk, I think that we should be offering them this opportunity, knowing that some patients who've received checkpoint inhibitors have had a response and didn't reject their organs. But this is a very dangerous thing to do, and the organ may be at risk. A heart transplant they did save. One of the ones in the literature when the case reports that got into trouble and they saved the heart transplant. But there you go, heavily immunosuppressing a person who's got squamous cell carcinoma again to try to rescue the heart. So this is brave new world, and we're in it.
So how efficacious is it at actually treating their squamous cell carcinoma?
So I think it's too early to tell. We're dealing with anecdotes. But here's the thinking. As I see these patients in the clinic and look at their faces. So the reason they're getting the squamous cell carcinoma is they've got an immunogenic tumor that they can't reject. So this should really work in transplant patients. But it's going to be risky for the transplant. So I think it should be more effective in transplant patients than in other people, because the reason they're at risk is that they've got an immunogenic tumor they can't reject. Otherwise, they wouldn't have the tumor. So I think that the chances of winning are high, but the costs and the risks are going to be a very difficult discussion in the clinic, especially at a time when we only have anecdotes. Yeah. But the invasive squamous cell carcinoma problem, if you're looking after 30-year patients, as some of you I know is getting to be a gigantic iceberg for us.
Ramesh?
So, I'm from infectious disease.
But you're okay.
I'm interested in immunology, so that's why I'm asking the question. So when you do these clinical trials, are you randomizing them into histology alone versus molecular methodology, this one, and see what the clinician responds to it? Because here we are assuming that the molecular, obviously it's clearly one step above histology, no doubts about that, but how do we assess sensitivity, specificity, or predictive values?
So these are observational studies, and they're prospective observational studies recruiting all the patients who consent, and we're not randomizing anything. Because I think what we're trying to do is get a feel for the clinician's view of he's getting readouts, she's getting good readouts from 2 platforms, and she's trying to tell us what the relative merits of the 2 platforms are. So we are not randomizing anything. Following that question up, some people have said, well, you should do a randomized study and see the effect on outcomes. So this has never happened, where people have actually shown that the test changes an outcome. So this is, if you want to set that hurdle, you won't have any tests. So right now, for example, Paul Terasaki's 1 lambda. So he started out with the microcytotoxicity assay in the early 1960s. And at that time, in the paper, you should read the Nature paper, he said, and at some point we should be able to do this with a Coulter counter. We should be able to have automated counting. And that ended up being the prediction of Luminex technology. So then when Luminex technology came in, it became a $1 billion company, but it never showed an effect on outcome. It was just a better test. And it actually was never approved by the FDA. It was just a better test. It's a laboratory-developed test. So when tests come in, the clinician will decide what the clinician needs. And the regulatory side is going to have to, we deal with the regulatory side, but basically it's going to be clinical demand. First, we have to establish clinical demand, clinical utility, but these are high hurdles. And if you don't cross the hurdle, all of this is of esoteric molecular masturbation until you get it into the clinic.
As a corollary to that, I am sure you have done body fluid analysis on your same methodology. Give us some insights on what you have identified using your own technology.
So we did a lot with blood. We looked at blood in the naive belief that the effector T cells would be in blood, but it's actually wrong. The effector T cell and the effector memory T cell are molecularly identical. So the recently derived effector T cell cannot be distinguished from the effector memory cells that you have a ton of right now. So we realized that even the idea of finding the signal in blood is incorrect. So that the transcriptome of the graft, the transcriptome of the lymphocyte, is not distinguishable in the blood, if that's the idea. And I'm going to look at the blood and find a magic molecule that no one's thought of before. That basically doesn't work. Basically, that's noise discovery. And there are papers that have said they've done it, but they're all noise. So blood doesn't have the signals, I think. The transcript signals. It may have other signals, but the transcripts it doesn't have. Body fluids has signals, but they're all non-specific. So they don't distinguish, for example, between antibody-mediated rejection and T-cell mediated rejection, because all you're seeing are the gamma interferon inducible chemokines, but they're present in acute kidney injury as well. So basically you're looking at the use of molecules in response to injury is very similar to the use of molecules in rejection states. So getting clear discrimination between these states in body fluids requires something like the tissue at the moment. It could probably be done in an observational study using machine learning methods that hasn't been done yet, but we're basically, if you're going to use an expensive technology to get non-specific signals in a body fluid, you then have to use the expensive technology once you've done that test. You now have to do the definitive test, and that actually is not good logic. Screening tests have to be really cheap. And in kidney, serum creatinine and proteinuria are very good tests. They're actually excellent tests. And you're going to have to do very well to beat serum creatinine and proteinuria by enough to justify a $1,000 test if you're doing screening.
I see.
So thank you very much for the talk. Very enlightening tonight. I think as you kind of pointed out before, you think about going to the liver side. And one of the things that we've encountered recently is that the higher incidence of what we suspect is possibly AMR. As you know, historically speaking, people who didn't believe in AMR, don't believe in AMR, and to distinguish that histologically is almost impossible due to the fact that you're going for secondary signs. C4D staining is not very accurate, obviously, in liver overall. And I'm curious to see, people have high DSA. And the biopsy from back says, T-cell mediated rejection rather than AMR. We treat them with T-cell mediated rejection, they get better. But do they really have an AMR component or not? We've been debating that issue for a long time. We've had about 5 patients we put on IVIG, plasma increases, whole works, not really knowing whether or not they really have it or not. And I wonder, I'm curious to see if you have preliminary thoughts on that.
Yeah, so liver, so in general, DSA is present. When it's present at the time of an indication biopsy, 50% of the people don't have antibody-mediated rejection. So now the DSA, as you know, is becoming granular. So there's complement binding DSA, IgG3 DSA, et cetera. There's MFI determinations. So the intensity, the characteristics of the DSA, they all determine the probability that's associated with the phenotype. So there's a lot more information we can be getting out of DSA, and that should be put into the equation. It has not yet. People are still thinking about DSA as positive and negative, and it's not. So DSA has a very high non-specific association with phenotypes, and that is makes the diagnostics that are overly dependent on simple DSA present or absent quite unreliable, as you've pointed out. We think that it should be quite straightforward to detect antibody-mediated rejection in the liver molecularly, but it's currently been a very evasive phenotype histologically, just as it has been in lung and in heart. So it should probably be a molecular diagnosis, and so we could make that. Treating antibody-mediated rejection in all the organs is very difficult, but if you can be confident that it's there, then that's the first stage in delivering the treatments which are effective. And there are clinical trials right now which are, I fervently believe that we're going to have labels in antibody-mediated rejection within the next 2 or 3 years. In other words, that insurance companies will reimburse, that you'll know that there's evidence that the FDA will accept, but you're going to have to know the disease is there. You can't treat these non-specific phenotypes and hope. So that, I really think that diagnostics, to move forward with clinically effective agents, you need effective, precise, accurate diagnostics to support that. And we're actually supporting now 4 different clinical trials for that reason. And finding some disappointing results. The molecular phenotype has not been changed by interventions, especially in this late onset de novo DSA phenotype. This is a very difficult phenotype to treat. And so far the results have been negative, but we're going to find a way of suppressing that.
Very interesting. Any other questions? Well, thank you so much, Dr. Halloran.
Thank you.